wanted to welcome you to the third webinar, Getting to Know Your Scientific Community. My name is Kirsty Bourne, and I will be your host today. Today's webinar is sponsored by my company, Elim Bio, and Elim Bio is busy supporting researchers on the front lines of COVID-19. During this unusual time, we wanted to make sure to feed scientific curiosity with those students that are home. We felt that the best way to do this is to give you at-home students a chance to learn some of the cool scientists in our community. And we have a very cool scientist today, and I'm going to put up a poll while I tell you a little bit about him. And you'll see a poll that's going to launch on your screen, and if you could go ahead and answer that, because that will help us understand more about what you know about what our panelist is doing. Today's panelist is Joe Solvason. And Mr. Solvason is a current Ph.D. student at UCSD. He's working to obtain a Ph.D. in Bioinformatics and Systems Biology. He does have a Bachelor of Science from the University of California, Santa Cruz, and his degree is in Molecular, Cellular, and Developmental Biology with a chemistry minor. He has had some really awesome experience working as a research assistant, including being a research assistant at NASA Ames Research Center. Mr. Solvason is a hard worker and a great researcher. We're very lucky to have him with us today. We will be learning more about his current studies and if he's able to have a work-life balance, being a PhD student. Before I turn the mic over, I wanted to do a little housekeeping. You'll see at the very bottom of your screen, there is a Q&A session. You can go ahead and type in any questions you have during the call, and I will go ahead and send those over, and we can answer them during any breaks that we have. We can answer them live, which is pretty cool. There's also a poll section, as you found out, and occasionally I'm going to pull up a poll, and if you could answer it. No one knows what you answered. There's no stress to get it right or wrong. It just gives us an idea of what your current knowledge is. And now, what we've all been waiting for, I'm going to hand over the reins, and we'll find out more about Mr. Solvason's research.
Hi, everyone. Hi, so yeah, my name is Joe. So I'm just gonna, is there a way for us to view the result of the poll? Will that be?
Yep, there you go. I'm gonna share them right with you right now. It looks like the question was, what is your experience and knowledge of cells? Looks like most people said, I have a good idea, but lack the details.
Good, perfect. All right, that sounds good. Well, I'll get started with my slideshow. All right, so are you able to see that?
We sure are.
All right, great. Yeah, so my name is Joe Solvason. I am a PhD student in Professor Emma Farley's lab at University of California, San Diego. I'm a student in the Bioinformatics and Systems Biology program, as well as the Quantitative Biology program. So today I'm going to talk just a little bit about like a high-level view, a broad understanding of the types of questions that we're asking and trying to answer in our lab. Some background on the topics, which allows you to understand some of the techniques that we use to answer those questions, as well as why we think these questions are important. So 9 months before you're born, you enter this universe as a single cell. And with time, that single cell is going to divide and divide and divide. And eventually you're going to, a familiar shape will appear. So this is an ultrasound of a fetus during pregnancy. And given enough time, these organs start to appear, right? There's the brain and the heart. And so one of the big questions that we ask is, how are the instructions encoded in this first single cell that enables the formation of a fully developed, complex, multicellular organism that we all are? So in many cases, scientists like to model processes that we don't know with processes that we do know. So I think this is an image that's familiar to everyone. So these are some baking ingredients. And so we can use instructions for these, which in this case are a recipe, in order to make a more complex form of these ingredients, which is just some bread. Similarly, if we have one cell, we know that the instructions that a cell uses is in the form of DNA. And I'll talk a little bit more about what DNA is. But the instructions for turning a cell into a multicellular organism are all encoded in this DNA. Now we're able to use the English recipe or the English language to read the instructions to convert these ingredients into bread. But we currently lack the ability to read thoroughly the DNA that's inside of the cell in order to interpret the DNA into like a logical set of instructions and how to make this transition from a cell to a human. So an example of a recipe might be measure yeast and sugar, mix, wait until bubbles emerge, mix in flour and sugar, et cetera, et cetera. Now, DNA takes the form of what you could think of as a sentence, a very long sentence, and all of the letters in the sentence are A's, T's, C's, and G's. Now, it's really tough for a scientist to just look at this and try and understand like what's going on here. What is it telling me? So to increase our ability to read DNA, it's good for us to think about what is important to include in any instruction booklet. Typically, you want to include what, where, and when. So going back to the recipe example, so step one might be adding yeast and sugar into a yeast bowl and waiting until bubbles form on the surface. The second step might be taking sugar, salt, and flour, putting it into the flour bowl, and do this while waiting for step one. The third step might be taking oil, stirring it into the yeast bowl, and after bubbles form in the yeast, and do this after bubbles form in the yeast bowl. Step four, you can take the contents of the flour bowl, whisk them into the yeast bowl, and you do this after oil is added. So this is so the point that I'm trying to illustrate here is that any set of instructions which you want to, which you're going to produce need to have what are the contents of this instructions? What are you working with? Where are you going to put them? And when are you going to put them? So going back to biology, I think there might be a fighter jet flying overhead. So let me know if you can't really hear me. So going back to this biology example, we want to be able to encode in DNA what to make and where/when to make it. And so we know that DNA contains the instructions for these. It's just tough for us to interpret this part. Where and when are we producing all the things that are encoded in DNA? So different things that are the different things that are that DNA codes the instructions for are making enzymes. So this is one example of things that are made inside the cell. And where the cells inside the stomach are going to be making these enzymes. And also things that are made inside the cell are these supportive structures, things that give you shape. And so these supportive structures are important in bones and muscle and other things that gives your body shape and allows you to walk around and navigate in the world. So DNA, regions of DNA which contain information on what to make are called genes. And the regions of DNA which contain information on where and when to make those genes are called, the technical term is called an enhancer. But for the purposes of this presentation, I'm going to call them switches, because what they effectively do is they allow you to turn on genes, turn genes on or off, depending on where you are in the organism. So if you are a heart switch, you're going to want to turn genes on in the heart. Whereas if you're an eye switch or a skin switch, you're going to be turning on those types of genes and those types of tissues. I see we might have some questions.
We do. One of the questions we have is from Logan, and he was wondering if there's different types of cells for different organs, or all the cells doing the same thing? Are they doing different things?
Yeah, so when we think about the way that a body is organized, so you have, let's take the heart, for example. The heart is a muscle that pumps blood throughout your body. And so there are a lot of muscle cells in your heart, and muscle cells look very, very different than other types of cells. Cells will specialize in order to do a particular function. But you could think about your brain, right? Your brain has a lot of neurons, which are the communicating cells which help you produce thought, and they give you this like consciousness. So there are tons of different types of cells. They look very, very different. They're very, very different sizes, but they all originate from that. Let me see if I can go back a little bit. But they all originate from that original cell. When you first emerge in this universe, you are a single cell, and that cell gives rise to all of the cells in your body. And so yeah, like I said, cells in the brain will be much different than in the heart.
It's pretty amazing, isn't it? Another question that we have is, what does A, T, C, and G stand for?
Yeah, sure. So A's, T's, G's, and C's stand for different nucleotides. So it's a little more. I can pull up an image later on if you remind me to, and we can take a look at what DNA looks like at the molecular level. But basically, all you need to really know is that the tools that your body uses to read DNA can distinguish between A's, T's, G's, and C's. And by reading the sequence of nucleotides, it's able to interpret how to make the different tools in your body, such as enzymes or supportive structures.
Do those nucleotides or cells ever get mixed up and end up in a different order than they should be?
Yeah, great question, and I'm going to come to that later on. Changes happen naturally. They happen occasionally. There's a lot of there are tons of molecular machinery inside of your cells, which sole job is to make sure that mistakes are not happening. Because when you go from one cell to two cells, you have to copy all of the DNA, so that all the DNA that was in the parent cells, you want all the information to be in the daughter cells. So when that DNA is copied, there's a lot of effort that goes into making sure that no errors are made. That said, when we think about evolution, what evolution really is changes in DNA over time. Sometimes those changes in DNA will be not advantageous, meaning it could lead to a disease or some kind of defect. Other times, those changes in DNA will result in no change in the organism. Then there's also changes in the DNA that can make you actually stronger for whatever environment you're living in. And that's what we think of when we think of evolution. If the environment can put a selection on a population, and people whose DNA gives them an advantage for some reason, maybe they're taller or faster or whatever, then they're able to go make it on to the next generation and pass their DNA on. These are great questions.
Yeah, we have one more, and then I'm going to let you get back to your presentation. Another question from Logan again is, do some of the cells not make it in, or some of the nucleotides not make it into the son or daughter cell in the process? Some of them get lost?
Yeah, I mean, I would say that there's many different ways you could answer that question, I think. There are many organisms which we've actually tracked every single cellular division. And so if you were to look at like if you were to look at this organism after it's divided so that there's 100 cells, we actually know every single one of those cells, and we can trace the lineage back to the single cell. And so that's called like stereotype division, where like you can, that process is 100% conserved for every like individual. Past a certain point, things get more complicated. I'm sure that there are like mechanisms in place where there's, you're just checking to make sure that everything's going right. Nothing's missing, nothing's in the wrong place. But what I would say is that our development from being a single cell into a fully adult human is very, very robust. And what I mean by that is that most of the time it works, which is really incredible given how complex it is. That said, things will always go wrong, and that's just life.
All right, we'll let you get back to your slides for a little bit. I'm sure we're going to have some more questions for you.
Yeah, and you're welcome to interrupt me in between slides if you want, if you think a question is good. All right, we know how to make bread. We know what genes and enhancers are. So I'm just going to talk a little bit about the specific questions we are trying to answer in our lab. So where are these switches in human DNA? So you can imagine, we know now, so in about in the early 2000s, the entire human genome was sequenced, which means we have now read all of the DNA. But just because you know what the DNA sequence is, so right, let's just say that in this part of the DNA, this is what the sequence is. Just because you know what that sequence is doesn't mean you know what all the parts are, how to interpret what this DNA actually means. And there are 3 billion letters in the human genome, or nucleotides in the human genome. And so just to give you an idea, if you were to write a novel and that novel were to have 3 billion letters in it, that novel would be 1.5 million pages. So there is tons of information in every single cell in DNA. So how do you make sense of all of these letters, these 3 billion letters? How do you know where the genes are, where the switches are? A lot of the work that has been done has been very successful in identifying where the genes are, but there's still a lot of work to be done, not just like where the enhancers are, but what I'm going to say in this next point, which is even if you know where an enhancer is in DNA, do you know where that switch will activate? Like which cells will, in which cells that switch will turn on? So if you have a heart switch, you want to make sure that that heart switch is only turning on in the heart. You would not want a heart switch turning on in the brain because, you know, all these processes are tightly regulated. And if you get genes turning on in the wrong places, that can lead to disease. The third question is tied to one of the questions that we got in the question and answer is changes to DNA can happen occasionally. And so we call each change to DNA a mutation. And we want to predict if a particular change to a switch will change where and when that switch activates. In other words, that is to say, if I were to change the DNA sequence of a switch, will that affect the behavior of that switch? Will that switch now turn on in the wrong cell type? Or will it turn on at the wrong time? So now I can go into a little bit about how we study switches. So we actually have DNA in the lab, and this is one part that I believe, one part of our lab that Elim helps us out with, is that we have DNA in our lab, which we've synthesized. And the DNA that we've created has a heart switch, and it's close to a red gene. And so basically what this red gene is when this heart switch turns on this red gene, you will see the cells which are heart cells. You will see them turn red. And so we study this construct, this DNA, inside of a model organism, Ciona intestinalis, which is just a fancy way of saying this is sea squirt. And just like a broad side note, scientists many times will study other organisms other than humans. And many times you're able to take what you find in those other organisms and apply them to humans. So other very popular model organisms to study are mice, rats, flies, worms, bacteria. And depending on what question you have, you may want to select a different model organism. There are some model organisms where the findings will be more applicable to humans, and there are other studies where you just can't feasibly do that experiment in that organism, so you have to choose a different one. So this is the adult sea squirt. It's about the size of your hand. It uses these little tubes here to breathe in and breathe out water and take in nutrients that way. But a very, very young sea squirt is going to look something like this. And so this is probably on the order of 500 or so cells. So it's very tiny. This picture was taken with 60 times magnification. But to make things a little simpler, I'm going to display this as a little cartoon here. So basically what happens when you put this DNA inside of this young sea squirt, you see red pop up. And it just so happens that this red region is where the heart cells are. Of course, there's not a heart there yet, but these cells, once in the adult Ciona, these cells will be the one which form the heart. So if we want to ask a question about this heart switch to try and figure out how it works, what we do is we take a copy of that heart switch and we make a change to it. And so the type of change we make doesn't really matter here, but the point is that you can make changes to these heart switches and then see how it affects how the switch works. So this is what we call a mutant switch, because we've made a mutation inside of the switch. So you could think of this as like, if there was a sequence ATTA, we change that sequence to AATA. So this figure on the left shows what we would expect for normal switching behavior. And now we're going to compare it to how this switch acts in this mutant C-squirt. And so there are some different possibilities here. Once we put this DNA in, it's possible that you'll see the red pop up in the same place as the normal switching behavior. And what that would tell us is that this change that we made does not change the way that this heart switch works. And so we might consider, and so another possibility is that the change that we make actually changes the location where you see red in the organism. And that is a really important finding, because that tells us that the region where we made that change is important in where the switch will turn on. It's also possible that making this mutation here, you might observe no red at all. And that's also an important result, because that means that that region that you mutated is very important. So by doing this process over and over again, not just making one mutant switch, but 2, 5, 10, we even do experiments where we have experiments where we can test 1 million switches at a time using a different methodology. And by doing all of these different experiments, we are able to formulate rules, hypothesize rules for how these switches are working. And using those rules, we can even predict, even if we haven't made a mutation yet, we can make a new mutation, and using our rules predict what the change in behavior will be. Do we have any questions?
We do. One of the questions is, do the switches ever create 2 changes at the same time in your body?
Sure. So this is just a really simple example. Yes, there can be very complex behavior show up. So there are tons of different cell types. There are hundreds of different cells, even in just this very, very small and this very young, small embryo. So when multiple changes happen, it's hard to interpret what's going on. So although it's really interesting when that happens, we tend to try and focus on making very, very small changes, which produce very, very small changes in behavior, because that way we can start to make one-to-one observations on, oh, if I make this change, I get this result. Well, if I make a different change, well, I get a different result. But if you have tons of different things happening, it's hard to interpret what's going on.
That makes sense, definitely. One of the questions is, do you sometimes have bad changes, some mutations that are negative to the organism?
Sure. So this DNA that we're putting in is actually outside of the DNA. It's kind of it's kind of an experimental an experimental detail. But since this is not actually required for proper development of the C-squirt, no changes we make here will result in a embryo that can't form. But you're totally right. So there are that's a really important experiment to do, is when you make these switches and they produce a different light signature, you should then go in and make those same changes to the organism and see how that affects the development.
That's great. Someone had a question relating to a virus, maybe something like the virus we're experiencing right now.
Yeah.
Would we mutate to stop a virus?
Oh, that's a great question. I think that would be really difficult. So what your question, that's a really important question, maybe not so much in viruses, but if any of you have heard of CRISPR or gene therapy, there are diseases out there where we know that those diseases are due to changes in the DNA sequence. And so there are efforts right now in order to, if we know that if we know that a baby will be born with a certain DNA which is going to lead to a disease, it's possible to go in and edit the DNA before, in order to make sure that they don't get that disease. Now, this also has like ethical implications, And it's a very, very complicated issue, and we're really nowhere near to having that play out in terms of editing human embryos, but you can even do this in human beings. So there are certain types of, I believe, anemias where we know there's like a single nucleotide that's been changed, and you can actually use biotech. We can use some very complicated molecular machinery to actually change the DNA inside the patient in order to cure them of their disease. But with viruses, this would be more complicated.
Yeah. I have a question specifically about the red gene control. If it changes the proteins that make the heart red, or does it just control the location where the gene acts?
Yeah, I think so. Basically, what this red gene is something that's totally separate from Sionna. So this red gene is actually just a fluorescent protein, and this is getting into the experimental details a little bit. But yeah, it will not affect the health. Us putting in this DNA construct will not affect the health of the sea squirt.
All right, cool. We'll let you get back to some of your slides. I have some more questions coming in, but I'm going to keep writing them down for the next.
Sure, that's fine. Yeah, so going off that red gene question. So this is a real image taken of Sionna, of a baby, of a very, very young sea squirt. And this is taken under a light microscope. So I don't know if any of you have been into any like school labs, but this is just a really fancy version of the microscopes that you see in any lab. Now, because of how fancy this microscope is, it has another mode, and that mode allows you to look for this red fluorescence, which will be, so wherever you see this red fluorescence will be where this red gene is being switched on. And wherever this red gene is being switched on, you know that it's being switched on by this heart switch. So if we turn on red mode, you can see this really bright signal here. And these three cells are the heart cells, are what we call primordial heart cells. These are the cells that are gonna become the heart in the adult organism. You can see there's kind of a red glare, like a red noise or a red shade over here, and that's just experimental noise. That just comes from the fact that this experiment isn't perfect, but we know that this is the signal. And so if you were to look at just sea squirt without this DNA that we put inside of it, you would not see this signal. And so this is another picture of Sionna. So I just wanted to put the second picture up because we don't only work with heart switches. So this switch controls a green gene. So you can see here that this green gene is active in these cells, and this is the nerve cord. And it's also present in these tail muscle cells. And it's also present it's very, very faint, but there's a very, very weak production of this green gene inside of the brain. And so we know that this switch, which is controlling the green gene, is active in the nerve cord, these two tip of tail muscle cells, and the brain. Yeah, so I guess are there any questions relating to those slides?
Let's see. I had a question about, how long did it take to make the formula to see red or green?
Okay, good question. So we got the red, so this red gene is actually taken from an organism. So these red and green and different genes are actually, so there are actually fluorescent proteins. So these red, we have a green gene, there are yellow genes and blue genes. And all of these genes are actually taken from other organisms. So we know the sequence of these because we observed, like in jellyfish, we see that they have fluorescence. So what scientists did like nearly 2 or 3 decades ago is that they were like, whoa, this is weird. These jellyfish are glowing. Like, what's up with that? And they found out that they were able to figure out what genes were producing that color. And some scientists down the line thought, wow, this is a really helpful tool. In this case, we're studying switches. a really easy thing to do is just a really easy way to see if the heart switch is turning on is to make that heart switch turn on a red gene or a green gene. That way you can really see like you can see where that gene is inside of the organism. So you can imagine that there are all of these different genes being produced in this organism, but you can't see any of them. And that's because they don't produce any light. This red gene, the only reason we're using it is because it produces that light, which makes it really easy for us to go in under a microscope and say, oh, that's where our heart switch is turning on.
That's great. We'll have another very detailed question, but I'm going to let you get back to your slides because I'm going to save it.
Okay, cool. This is the last slide, just talking a little bit about why this research is important. I've kind of touched on these throughout the presentation, but I'll just kind of sum up here. So switches are required for correctly producing the right ingredients or genes for your body at the right place and at the right time. So making sure that heart genes are expressed in the heart, making sure eye genes are expressed in the eye, and so forth. Therefore, changes to switch DNA can cause problems in the body. Well, if these switches are so important in coordinating what genes are expressed where, if you're changing these switches, there could be changes to what genes are expressed where. And so that can lead to disease. So understanding switches, how they function, what breaks them, will allow us to predict who is at risk for diseases. So I don't know if any of you guys know the company 23andMe, but this is a related effort. So what they're trying to do, the service that they offer is that you take a swab of your mouth and you can send it off to them and they can tell you what your DNA sequence is at regions that we know are important. And so what this can tell us is, say like if you're at risk for diabetes, this can help you make lifestyle choices to make sure that you really have a good diet, that you exercise often, because those are going to be activities that are going to lower your risk for diabetes. And understanding these switches also allow us to create new treatments. The next generation of medicine will likely include a more of a bulk of what's called gene therapy, which I mentioned earlier. And so basically what gene therapy allows us to do is if someone has a disease which is caused by changes in their DNA, you can actually go into their DNA and change their genes back to normal. Currently the uses here are very limited, and that's due to a lack of technology. But in the future, our technology is going to be better, largely from all of you guys going to grad school and giving us tons of new technology to work with. And that's going to enable new treatments and be able to treat more patients. That's it.
So one of the questions was actually related exactly to what you were saying, so I'm glad I saved it. The question was, what's your prediction on how close we are to be able to make the changes in DNA in humans for the positive change to decrease the negatives?
Yeah. So I think there's two ways that you can make there are several ways you can make changes. The first way is by going into someone who's living, an adult. You give them the molecular machinery necessary to make the changes in the DNA that you want to change. And that is already happening now to some extent. Unfortunately, the tools that we make, so the molecular machinery we use to make those changes in DNA, they are not just made out of thin air. We are not just writing the DNA sequence. What we do is we learn from organisms and we take bits and pieces from all these different organisms and we combine them to make a functional tool. So CRISPR, which is a tool which allows you to say, oh, I want to change this DNA sequence from this sequence to this sequence. We actually got that tool from bacteria. And right now we're using, so when the AIDS virus came around, a lot of scientists were working to cure the disease, obviously, but there was also an observation that we could use a version of this virus, which is not harmful to humans. We can use it to modify the virus to make it not harmful, and then we can use that virus in order to introduce new genes, new beneficial genes into humans. So if you have a defective copy of a gene, you can actually use a virus to deliver the proper copy of that gene, and then the virus will just die off. So a large part of this, so how close are we? I think we're already there, but there's still tons of improvements that have to happen. The other way of making changes to human DNA is by doing it before the baby is born. And that's a very, very controversial topic that I think the scientific field hasn't really settled on if it's ethical. And if it is ethical, how do you go about doing it in a ethical way? Someone has actually already done this in China, and you can look up the news reports online. But it was kind of a rogue scientist who just took it upon himself to do this. And so we really have to be careful when we get increases in technology, it really opens the door to things that we can do. And so we have to be very careful about what kind of changes we're making.
I have another question. If you're done with your slideshow, do you want to unshare and so we can see you?
Yeah, sure.
Okay, perfect. When we change a gene, does it stay changed forever?
Yeah, so that's a great question. So it depends on if the change is made in your germ cells. So basically there are cells in your body which are used to procreate. The first cell that arises when you come into this universe is one cell from mom and one cell from dad. If the change is made in these cells, then yes, the baby will get that change. However, if I make a change to my DNA in my hair for something, or my eyes, then that change will not be inherited by my child. But that change will stick with me in my eyeballs for the rest of my life. Yeah.
All right. Wow, it sounds like you have lots of research you're doing. Do you ever have time to do anything else besides research?
Oh, what a great transition. Yeah, so I try to keep a good work-life balance. Grad school, I think, if you ask 5 different people what grad school is like, you'll get 5 different answers. For me, grad school, I really cherish that like work-life balance. So I work from roughly like when I wake up in the morning, which is like 7 a.m. I'll get like started around 7:30 or 8, and then I'll work until like 5 or 6. And I really enjoy the work that I do, so it's not really a taxing thing. On the weekends, I try not to work at all. But there are like maybe one weekend out of every 4 weeks or 5 weeks where I just have things I need to do. Like if I'm giving a talk, I'll need to prepare for that. But for the most part, I try and keep my work during my working hours. And I've been fairly successful at that so far.
Wow, that's cool. That's awesome. So you still get to have fun with your friends.
Yeah, exactly. Yeah.
Oh, good. Well, that's good to know.
Yeah, I think that, yeah, like my boss also knows this, and she has told me this like explicitly, which is like you need to make sure that you take care of yourself always. So if you're only working, you need to have a balance to rest. So you work all day, you don't want to work all night because you can't just like wake up and then start working. And it's really good to have like that off time.
Yeah, yeah. What does your lab look like? Could you explain it a little bit to the?
Yeah, I actually took a picture of. Let me see. I just grabbed a picture on Wikipedia on Google Images of what a lab looks like. This is like one bench of a lab. So let's see if I can present. So you can see here the scientist. There's like some refrigerators. It's really nothing special. Like these are just white walls. You just have like a black desk. It's very, very long. There's like tons of stuff on it. Our lab is a little more organized than this, but like typically when you go into a lab, it just looks like a normal office space, except for there are very, very large benches where you can do very, very large desks where you can do all of your lab work. But in some places in industry, like they have very, very fancy looking offices, very, very fancy looking labs, like what you would see in a movie. But most academic labs are just like, they're nothing special, but yeah.
Well, the magic happens there,
Yeah,
though.
exactly.
And I know your lab's at UCSD. Can you tell everyone, if they're not from San Diego, what that kind of looks like at UCSD and where we are in relation to other things in the city?
Yeah. Let's see. So what do you mean by that?
Because I know there's people on the call from the Bay Area and from other parts of the country. They might not know, where in San
Oh, yeah.
Diego the university is in location to other markers. And
Yeah, so San Diego itself is located in La Jolla, or the UC San Diego is located in La Jolla, and our lab is located near the hospital because our boss is appointed through both the biology department and medicine. Um, it's we're La Jolla is kind of not a very big. It's more of just residence for students and the families that live there. So there's nothing much going on. But if you drive like 15 minutes south, you can get to all the different beaches that are there. And if you drive a little further south, you can you hit the downtown area, which has a very nice nightlife. But I typically stay up here more nights. Yeah. But there's also a lot of like really cool outdoor things you can do. There's like Joshua Tree 2 hours away, so a national park. Las Vegas also has a few like state parks that's like 5 hours away. So it's definitely nice living down here.
that's great. And how did you become interested in science? Were you raised always in science, or did you have somebody that inspired you?
Yeah, both of my parents are scientists. So they met while they were in graduate school. They never really pressured me to become like a scientist or go to grad school, and they were very adamant that they did not pressure me. They remind me that. But I really, it was always on my mind that it was a path that I could go down because they had already treaded that path. I knew that you could be paid during your PhD, and so I knew that it was a viable thing to do. I entered college as a psychology major. Then I switched to marine bio, and then I switched back to psychology, and then I switched back to molecular bio, and I ended up sticking with that. I really got interested in the sciences. Like I got more interested around my second year. I did a 2-year internship, like you said, at NASA in a chemistry lab. Maybe idle is the wrong word, but they're just hilarious. They had such great dad humor. They were just funny guys to be around, and a great group of people to just spend my time with. It was really interesting talking about science and thinking about science. And one thing I learned during my time there and at undergrad was that I don't think I could ever work in business or like the humanities or like philosophy or like politics, because I think humans are really complicated. Like their behavior, it's like just hard to know. So it's nice when for me, the sciences are nice because you're just in pursuit of one thing, and that's the truth. And like, if you end up being wrong and someone proves that like what you said isn't right, that's fine. And you just need to like build off that. Yeah. But I feel like the scientific community on the whole is really the goal is to like figure out what's going on. And it's less like these gray area arguments. But yeah, I didn't really know coming out of undergrad. I wasn't really sure if research was right for me for like the long haul. A lot of people gave me the advice, don't go to grad school until you're like pretty sure that you want to do research, because it's normally like so my program is normally like 4 to 6 years, which seems like a long time. But if you're doing something that you really enjoy and you're being paid for it, like I didn't really, it's a pretty good a job, I think. So
I think a lot of students don't understand that it's possible to get paid during your PhD.
Yeah. I mean, when I got out of school there, like, I think your early 20s are really rough. Like once you get out of undergrad, you need to figure out like, what job you're going to get and all this stuff. And so I was always very low stress in that area because I just worked as a research assistant for a few years, and I was like, you know what, I really like research. I would like to try to just apply and see what happens. And I think one really important thing to keep in mind if you are considering graduate school in sciences is to just get some internship experience at a lab, at your university, or wherever. Those opportunities are actually like not very hard to find if you know where to look. So like just reaching out to like your student counselor, or even just emailing a couple professors at a university saying like, hey, I'm really interested in getting into the sciences. Like, how do you think I should go about this? Like, I think you'd be surprised at how enthusiastic scientists are in getting people involved who are motivated to do so. It's not always about like who was in the best math class growing up, who was, who has all these accolades and honors. Like, I think motivation, showing that you're motivated, showing that you really want to do it, goes a really long way in science and academia. So I would definitely encourage you guys to not feel intimidated by the sciences, because I had no idea what doing like research was like in my undergrad. And like, when you're in your PhD, you're supposedly pushing the bounds, like you're doing things that no one has done before, but you're also supported quite a lot. So there are people who have already completed their PhDs who you work with. There are professors who are there who have their own ongoing projects. Like, you're totally supported the whole way. And a big part of your PhD is becoming independent, being able to do science independently. But they certainly don't expect that when you're an undergrad. They certainly don't expect that when you're first starting your grad school. That's why you go to grad school, is to learn all these things. And you get paid to do it. Like, you get paid to go to classes and stuff like that. So, and when you get out, it's like, it's a really great degree. Like, I know that in 5 years, if I am sick and tired of research, that's okay. There are tons of other opportunities that a PhD allows you, that I'm not stuck in the sciences if I don't want to. But I know for the next couple of years, I'm going to be really excited to work with the people in my lab and my boss to try and add some knowledge to what we know.
Yeah, I think that's really the best part about science, is how everybody kind of, we're all searching for truths, and everyone helps each other. It's pretty cool.
Yeah.
Has anything changed with you, with your work-life balance, with your lab, with COVID-19?
Oh, yeah. Definitely more on the side of less work due to COVID, but and that's mostly just because of working from home. It's just, if I could go to a coffee shop every day, I would definitely be more productive. But yeah, so I'm in a bioinformatics program. I do some experiments, but for right now, I'm just doing, since I'm at home, I'm able to just work on the computer. Mm-hmm. Yeah, so I'm able to do all of my computer work from home. I've been reading, I've been doing like some statistics. I've been helping out another student in the lab with like some statistics on their projects. So I've definitely been keeping busy. And that is one thing that my boss is also good at, is making sure that that I always have something on my plate, and I don't feel like I'm floundering or anything.
That's great. Well, I wanted to thank you so much. We had quite a few people that just responded, and thank you. We even had somebody wondering if you would be able to share your presentation with other companies or colleges or globally. So I'll find their email and I'll connect you too, if that's all right.
Yeah, definitely.
Okay, sounds good. I wanted to thank you so much. I appreciate you sharing your knowledge and your time, and just taking the time to inspire our students and help them know what it's like to get your PhD. Because it's hopefully some of them on the call are going to be thinking about that later.
Yeah, definitely just keep an open mind. Like, just take some science classes. And if you think you might be interested, just like send emails to people. Like, I think you'd be surprised at how encouraging scientists are.
That's awesome. I agree, definitely. Well, I wanted to tell everybody a little bit about our webinar next week. So next week, our webinar is going to be on Friday, April 17th. It's at 1 o'clock again. And if you have any questions about COVID-19, this is the place to be. We're going to have our panelist on Friday is Dr. Mikhail Tal, and she's the infectious disease team lead at Stanford University. And Dr. Tal has her PhD in immunology from Yale. and she'll be explaining her current work, just like we heard today, about your current work, Joe, and then we're going to be having time for questions. So if you have any questions about COVID-19, make sure to watch your email on Monday. I'll be sending out the link for that. Joe, thank you so much. We really appreciate your time, and we really appreciate your knowledge. And friends, as usual, make sure you stay kind, stay curious, and wash those hands. Thanks, friends. Bye.
Bye.